In the previous section, I made a small error. The number of naturally occurring amino acids are 20. This is a correction. This depends on the side chain that we can classify them into two groups of polar or non-polar. This is a typical table you will find in most molecular biology textbooks, and this one is from Bruce Alberts and company. So 20 naturally occurring amino acids, not 24. So we move to another important biomolecule. We had talked about the size of lipids. Now we will talk about the size of DNA. And please remember that I keep insisting on nanometers because most biologists study the rise per base pair as 3.4, and then they don't remember the units. Since this is 0.34 nanometers, it should tell you that 3.4 refers to angstroms. But in SI units, we're talking about 10 to the power minus 9 meters is 1 nanometer, and 0.34 into 10 to the power minus 9 meters is the rise per base pair. You might also be benefited by remembering that the diameter of the double helix, if you treat it as a simple cylinder, is 2 nanometers. So each brick is layered at 0.34 nanometers and the diameter is 2 nanometers. Could say the radius is 1 nanometer, depends on how you want to think about it. We're all familiar with the bDNA, and that is the most commonly occurring DNA, which has a major and a minor group. Depending on environment, pH, salt concentration, you may find A or Z forms. We're not going to worry so much about the less common forms and assume for our further calculations. Remember, this is quantification and order of magnitude estimates, biology by numbers. So we're going to assume that it is bDNA largely. The amino acid genetic code is connected, the amino acids are connected to the genetic code through this beautiful representation, the P. These tell us that as we go from inside outwards, for example, the start codon methionine is AUG, yeah, and many of you studying molecular biology know this, AUG. You can also AU, A, C, and U. You can also find these producing isoleucine. So, in a sense, we know that a combination of triplets A, G will both result in lysine. This is the redundancy. And some amino acids like G, C, and then U, C, A, G are encoded by multiple such redundant codons. This codon to amino acid forms, in some senses, the crux of the genetic code. It's the secret language of genes and proteins. So triplets of nucleotides are a codon, and they encode in a redundant manner for amino acids. Some of them, like UGA and UAA and UAG, encode for stop. This is translational stop. There should be no more translation at and beyond this point. In a way, it's like learning a language. The Barakhari of Devanagari or Urdu or Tamil or Kannada or Telugu or Homia or any language, German, Spanish, Italian, Tagalog in Philippines. The point is that if you want to study molecular biology, you need to understand at least the basis of the genetic. And if you spend a lot of time in it, you usually end up remembering it. The standard ruler of E. coli, which we said is a very well understood organism, is also easy to isolate, possible to do genetic manipulations, and physical principles are in common with more complex cells. The biological macromolecules that make it up consists of these A, B, C, D. And if I ask you to guess what these are, it's probably easy for you to figure out that A is DNA, BDNA in fact. B, it's a protein. It's a large protein, in fact, as you can probably see. It's oxyhemoglobin, consists of the tetramer. C is a lipid, phosphatidylcholine. And D is one of those molecules, for macromolecules, whose biochemistry is even less understood than lipids, namely polysaccharide. This is a M41 capsular branched polysaccharide from Escherichia coli, the bacteria. These structural representations are important because they tell us the atomic positions. But our goal in this this idea of quantification using rough approximations is that we use the approximate size scale to get an idea of how things relate to each other, to then develop an intuition. So DNA, in fact, especially in eukaryotic cells, is considered to be organized at multiple scales. So we are familiar with the 2 nanometer diameter and 0.34 nanometer rise per base pair DNA helix, B size, B form. And at its simplest level, it is this that forms the chromosomes, even in human beings. But it's not available just as naked DNA or plain DNA, right? It is organized around nucleosomes. These nucleosomes form a core of 8 histone molecules, around which this is wrapped in a 1, 2.5. Yeah. So 1 is completed, 2 is completed, and 4. At the a clip kind of molecule that acts on these histone octamers is the H1 histone, or sometimes called the linker histone. The diameter of such a histone complex with DNA, the nucleosome as it is also sometimes called, right, is 11 nanometers. It forms a sort of cylindrical geometry and is separated by a length of DNA. So it's 1.65 times, sorry, not 2.65 times, 1.65 times. The linker DNA that joins each histone is thought to be typically an average length. This then wraps into a more complex structure, which forms a 30 nanometer filament, and it's called that because the diameter of that filament is 30 nanometers. That in turn forms loops averaging 300 nanometers in length, and these are then loops, sometimes called giant loops, which then themselves are organized into 250 nanometer wide fibers, the thickness of these fibers, with a diameter of 700 nanometers. So we went from 2 to 700 nanometers. Yeah? This is what the form that we can potentially find chromatin in, finally of 1.4 micron diameter for the 2 arms of a chromosome in mammalian systems. That is really astounding. There is such a high degree of compaction and arrangement and resolution of fine structure that it would be amazing how anything works, because if it is so well organized, at the time of division, at the time of mitosis, the chromatin decondenses and loses most of this organization and becomes loose. We think it is either in this state or in 30 nanometer state. And it reassembles again. So this cyclical assembly and reassembly is a very important thing in cell biology. The length of the DNA is of 146 base pairs. You could round off to 150 base pairs. There are two each copies of H2A, H2B, H3, and H4. That is the octamer, 1, 2, 3, 4 into 2, 8. And it is linked at the exit points of the 1.65 rounds by a H1 histone. Diameter 10 nanometers. And if you assume that this is 2 plus 2 nanometers, then it's 4, maybe about 6 nanometers in height. These numbers are useful for calculating simple things about the number of nucleosomes and the size of nucleosomal DNA as compared to plain DNA. As far as cells go, so far we've talked about the cartoon representation of E. coli. Now let us talk about a cartoon representation of a more complex cell, a larger cell. This is the fibroblast. This fibroblast, I said earlier, it looks like a pizza slice. It has cortical fibers, which are made of actin, that extend into these extensions called filopodia and lamellipodia. They have a bulging nucleus in the center, and it kind of forms almost a two-dimensional structure. So you can approximate it as a two-dimensional structure that has a bulging nucleus in the middle with a diameter of 10 microns, and one side of this triangle is 50 microns. This means that we can calculate the approximate area and even volume of the nucleus as well as the cell. So now we can ask the question, what volume fraction of the human genome forms the volume of the cell nucleus? How much of the cell nucleus is occupied by the genome? To do that, we must know the approximate size of the human genome. So you need to look this up, but L human genome is, and I remember the meaning of this sign, it's an approximation, is 10 to the power 9 base pairs. Each cell has this much DNA, right? We said that 146 base pairs is associated with one histone. So we call this 150 base pairs. The diameter of such a structure is around 10 nanometers. The length of the 146 base pair DNA is 146 into 0.34 nanometers, which comes, let us look at our calculators, 146 into 0.34 is 49.64, so I'm going to say 50 nanometers. So we have ten to the power 9 base pairs into 0.34 nanometers divided by 50 nanometer segments, which gives us 6.8 into 10 to the power 6 segments. If we say that the volume of this is the volume of a cylinder, which is pi r square h, and the height is 6 nanometers, then our genome volume is the number of segments into volume of the nucleosome And we want to know the ratio of this to the volume of the nucleus. I think you have understood the method by which we do this, and I will expect you to solve this on your own. We now come again after DNA to proteins.